So good morning, everyone. I'm going to tell you about the ways and methods and purposes behind some of the mapping of mysterious microbes that we've been doing in my laboratory and also with a large consortium of other collaborating labs all over the world, and also the technology that's made this possible. So this is also my Twitter handle if you want to follow. Also, we tweet about all kinds of things. It's also generally a good repository of things I'm reading or thinking about. It's also, Twitter, as most of you know, is a major platform for presidential policy announcements. So it's a very official platform. At least it has become so. And so it's also a good place to look at updates from the lab. So I want to start quickly with this chart, which is basically the history of how much it costs to sequence one human genome. It used to be, if you remember the first human genome project being announced, or you were probably reading about it, was in 2001. And also, you know, there was updates to it in the subsequent years, but it used to cost hundreds of millions of dollars to do just one human genome. And due to a lot of competition and innovation in technology for sequencing DNA and RNA, it suddenly became much cheaper in this time period where basically every 5 months the cost to do an experiment got cut in half. And this was a wonderful time to be working in genetics because it was almost impossible to over promise what you might do because the cost kept coming down really fast. And it's plateaued a little bit, somewhat from the market trends here a little bit, from Illumina as one of the big sequencing companies. But basically it means that, you know, now it's about $1,000 to sequence a whole human genome, which you can then embed into medical care. So for example, at Weill Cornell Medicine, at New York Presbyterian, almost every cancer patient who relapses, we look at the tumor, we sequence the DNA of the tumor, look for mutations that we can target, and then have genetically driven and genetically guided therapies for patients at the hospital as a way to understand it. And so instead of it being exorbitantly and impossibly expensive, it's now something that's not even that much more than the cost of an X-ray and a radiologist sort of interpretation. So it is on the hardware side, and just the cost of it is come down pretty low. There's even some rumors that sometime soon we'll have a $100 genome from Illumina, is one of the biggest sequencing companies that does this. And so what's extraordinary is because of this chart, it means every day we're generating more data than ever before, and it's cheaper than ever to do it. That actually means that every single day that you see me, if you see me in the elevator, you see me like wandering around the streets, every day is the best day to be a geneticist. It's by far the day with the greatest potential for generation of data, the most interpretation and analysis you can do of data. It's literally the best day of my entire professional life. Every day you see me. Now, it doesn't mean every day is the best day personally. Like sometimes I spill coffee on myself, or my daughter says something weird to me in the shower that makes it think like the shower's leaking. Anything could happen on any given day. That doesn't mean that every day is the best day personally, but at least professionally, that is very much the case, because we can sequence things much faster and much cheaper than ever before. But there's more than one genome. So I was talking a lot about the human genome, but you have to consider, of course, there's more than one genome. So in particular, if you were to imagine what does the world look like from a microbial perspective, you can see here that this is, droplets of air surrounding us. You can see we have bacteria, viruses, fungi, spores all around us, and the surfaces from all over the, every room we're in. You can see here that you leave a handprint that has a microbial residue. Every time you move your hand, you'll see microbes move around. Every time you look at your skin, you look down at your hands, there's this entire microbial ecosystem on your skin. Every time you breathe out, you'll have aerosolize also some droplets, which I think a lot of people have been thinking about, especially in COVID, and I'll come back to that. You can see here in this picture, this is the sort of gut microbiome. On the chair is a microbiome. You can see your mouse and keyboard, your computer have their own microbial signature left behind. If you look in a cup and just like look at what's the bottom of a cup, you put a pen in a cup, it'll get a poof of microbes that come up every time you put that in. Some people think this video is absolutely disgusting. I think this video is absolutely amazing. You can see here, even the buildings in which we live and work have their own built environment microbiome, which I'll tell you about this morning, is ways that microbes move in and out in between different buildings. And so what this means is if you think about your cells, you think about them, there's these human cells, but we have this entire ecosystem all around us of these microbial cells. If you were to step onto a scale, several pounds of your body is actually made from microbes. So if you are mad about your diet, you can actually blame, in some cases, the biochemistry as well as the weight of your microbes. And that's because they serve as this entire pharmacy, this entire ecosystem in you that is processing actually a lot of your own biology and creating a lot of the biology. So for example, estimates from Lee Hood think that about a third of the small molecules in your blood either are made by the microbiome or processed by the microbiome. And this number has fluctuated in different studies, but it stayed pretty constant. And so if you think about what's in your body, it's not just about the microbes and human cells, but it's this entire biochemistry and interaction between them. And so this has led to really large-scale projects and excitement called the Human Microbiome Project, excitement to understand these microbes. The International Human Microbiome Consortium is one. There's also links to obesity, to how you process food, links to inflammatory bowel disease. We recently published clinical trials that just show the impact of probiotics on inflammatory bowel disease. So there's a lot of power in looking at, these interactions between microbes and human cells. It's now actually over a dozen different diseases that have been linked to the microbiome. So some of these make perfect sense. So for example, eczema, of course, ulcers, think of Heliobacter pylori, you've probably heard of, but things like autism or cancer or depression, which were really, completely outlandish 10 years ago, are now mainstays of funding from the National Institute of Health to look at the, interaction between what's happening in your gut, what's happening in your brain. How do we have a more holistic view of what's happening for the biology in your body? It's also now kind of standard teaching for medical students as Heba can tell you at Cornell. And that's because it does, it's been linked to a lot of these diseases, but also it has really surprising features. So for example, there's a beautiful paper from a couple of years ago by Strassman and Todd Gullab that showed that basically the tumors can have and harbor bacteria inside them that can also mediate resistance to chemotherapy. So you maybe have to first treat with an antibiotic before you do chemotherapy, again, showing that there's this really intriguing connection between the bacterial sort of biology and also what's present for the human biology. So these are continual exchanges of molecules and regulatory processes. And, they're not just resisting things sometimes, they actually, can even process them differently. So for example, this is a paper a couple of years ago also that showed about a quarter of the drugs that have human targets. So again, like antidepressants, you'd think, can you make your bacteria antidepressed? You would think no, because they can't be depressed, but they can be inhibited in terms of their growth or their biology. So this is another just great example of, you think you're targeting one biology, you're actually targeting, multiple kingdoms and domains of life at the same time. As Pierre Bourque, an old guy I collaborated with back when I was a postdoc. Really great paper there. And then my other favorite papers from Mohammed Dania and Michael Fishback that also showed that, if you look for biosynthetic gene clusters, which are these clusters of genes that compose one biosynthesis pathway, if you just look in normal human flora, like for example, look in the vaginal microbiome, these are just normal flora that would be present in healthy people, you can actually find new species of bacteria and even predict novel biology, new biochemistry, in this case, a new antibiotic that was being made by these flora. And basically, these organisms work on your behalf. You don't have to ask them to do it. They'll just do it because they're really just here to help you, or they're helping themselves mostly, but it happens to help you as well. And so it's really also exciting to think about, how you have these, microbes that will do you a favor effectively without you even have to ask them. So you're kind of self-medicating in that way. So all this work together as a background means that if you're a human geneticist today, which I was originally trained as, it means you're actually a very bad human geneticist if you only look at human DNA. So to actually be a good human geneticist, you have to not just think about the human DNA, but you have to be kind of a cross-kingdom geneticist or cross-domain geneticist without a bias of kingdom. And so, this is because of all the reasons I just showed you, but it's also because many areas of the world have, of course, even more peculiar biology than just in your own body. So for example, one of the projects in the lab is, and that some of you will be looking at some of this data, is the Extreme Microbiome Project, which looks for microbes in strange places to understand what they're doing. So for example, this is a lake in Australia called Lake Hillier, the pink lake, and it looks actually like this. So if you go to the lake, you'll see this sort of candy pink lake. It's very salty. It's actually, if you put your feet in the water, your skin will burn because the salinity is actually lysing the cells on your surface. So it's very painful to walk through. There's a great picture from Ken McGrath. And like if you go to the shore, you can get huge chunks of salt. They're just wandering on the shore. And it's actually Daniela salina is one of the organisms that's in this lake that actually makes it so pink. And there's algae, there's different microbes in this lake, but we know very little about them until we've been able to sequence them and start to catalog them. And it's not just here. There's also places like in Antarctica. There's a friend, collaborator, Scott Ty, who went down to Antarctica and did sequencing in the middle of Antarctica and got that set up. We've also looked at places like in New York City, the Gowanus Superfund site, and that's a Superfund site. Notice the D, not a Superfund site, because it's totally full of toxic sludge. And so there's many places that we've been sequencing and begin doing annotation and curation of different extremophiles, so extremes in consequence of salinity, temperature, pressure, gravity, in this case of the space station, or where they have really high or low pH, or even ions going through. So, this is part of the raw exploration part of it. And so this seems to be kind of an abstract and intriguing sort of direction where microbes can persist. But I think this used to all seem very abstract to almost everybody that we talked to until recently. The idea of tracking and monitoring specific microbes, in this case, thinking about, for example, SARS-CoV-2, which is now killed over 120,000 people in the U.S. and infected over 9 million people globally, is a much more, I think, concrete example of the risks of things around us, and also the fact that you need to be able to trace organisms to know what they're doing and where they're going. And so, I think most of you have been thinking about this. You're all probably sitting at home because of this. But, this idea of what's on the subway, it used to be, people used to do, before there was masks and gloves, people would do things like this. They would just take a used paper towel as a defensive shield against touching this steel railing. Or in some cases, I'd catch people wearing lab gloves. And so actually, in the subway yesterday, when Heba and I were collecting samples, it was the first time we'd gone out and saw lots of other people, with gloves or masks on. But in years before, of course, it was rare to see it. And so this was often done because people had no knowledge of what was there. There was complete terror in the absence of a pandemic of what was there. But realistically, there was no knowledge. So when I took these pictures back in 2010 and 11 and 12, and I was always curious about what was there just because I ride the subway every day back and forth from lab. And I'm, live in Brooklyn, so I often go around to other places. But I also became very curious about what's on the subway because when my daughter got old enough to ride the subway, you can see here I shamelessly use her in my talks. She got old enough to ride the subway, and there was one day when she did actually grab the subway pole, and I didn't get a picture of it, but she licked the subway pole, and I became completely intrigued / terrified of what exactly had just occurred. Now, clearly there had been a microbial exchange of some kind. If any of you have been licking public surfaces, you would know this as well. But you would always want to wonder, well, what exactly got exchanged? What's on this pole? What's the microbiome of the mouth? How much do they move back and forth? Should I be concerned? Do I, run my daughter home and try and throw in the shower? What do I do? Does it matter what's there? And there was actually no information. So some of the people on the call, like Abraham, helped get a lot of this project started and led it back in 2013, was to say, well, we don't know what's there. So, in the absence of knowledge, I think the best thing to do is to explore, is to discover what you can find about the reality that's around us. And so we launched a project to look at the microbiome of the metropolis, or what the New Yorker called the metropolome, to see what's in the world, all around us. And so in particular, what we did is went out and said, if we're going to do this, we're not just going to do 1 or 2 boroughs. We're going to do all of them, all 468 subway stations in triplicate. A little bit of formatting issue here. We're going to annotate them and upload it and then timestamp them, and then brought them all back to the lab. Got about 1,500 samples brought back to the lab, sequenced them to get 10 billion fragments of DNA, and then take them all and map them to all known species. And still one of my favorite features of the study is that when you take a look at all the DNA, on average, about half of it matched no known organism. We'd never seen them before. We don't know what they are until we did the study. And a lot of them were bacteria and some viruses. I'll tell you about these shortly. But we essentially had this extraordinary unknown biology right under our fingertips, and half of it was unknown. And we could see some species that we would make plots of, but you can see here, but most of it was unknown. And so this was really intriguing. And then we thought, well, of the things we can see, where are they and what are they doing? So for example, in the Gowanus Canal, this looks like a place that you shouldn't swim. Here we can actually see these, basically methanobacteria and other sort of, essentially bacteria you'll find in sewage would also show up there because we know there is some sewage there. And you might think that sounds gross. Would you ever swim there? It has been recommended by a guy who swam in there that you should not swim in there, is what he said. He tried it in a hazmat suit and could not make it for the whole length of the Gowanus Canal. But the other things we, want to think about is not just the known species, but also these unknown organisms. Is this ongoing and continual question is, who is there and what are those species making? And so for the known species, we could start to build some of this out, and we could see where they occurred. So for example, there's some Anarcoccus, some Staph aureus, some Pseudoaltimonis that were in different places. But this one, as you can see, was very distinct. This is one station. It didn't appear anywhere else across the city. And this actually was the place we worked with the MTA before they opened the station back up to the public. This was flooded by Hurricane Sandy. And actually, we were able to see these 10 species here that are unique that we didn't see anywhere else in the subway system. And so if you remember, I don't know if you remember, Hurricane Sandy came through. It knocked off government websites. It really made huge waves in the city, giant waves coming through. It also, some parts of the city got huge amounts of water. I don't know if you remember this, but this is I'm just kidding. This was the day after tomorrow. It wasn't quite this bad. This was not quite that bad, but it did feel that bad. I remember I still had tape on my windows for years afterward. But this is the actual map of the transit system. You can see here all the lines in gray were the transit systems that were either inundated with water or inactive or actually submerged in water. In this case, for example, South Ferry Station. So we worked with the MTA to get early access to this station and asked that question of, well, could we detect this molecular echo of the cold ocean water, because you can see here the whole station was underwater with this cold salt water, and what was there. So this was really intriguing, and some of the species we found were previously thought to be Antarctic species, like Schoenella frigidmarina, that has an ability to produce eicosapentaenoic acid. And which is interesting because, where is this normally found? This is something that you'll find often in the human diet by eating, fish or cod or, salmon. So this is normally found with fish, which is intriguing. And if you have low levels of this in your diet, you actually would be at a higher risk for suicide. So it seems like if you did happen to lick a pole or drop some food and get this in your mouth, it wouldn't necessarily be a bad organism, certainly not a high risk one. And so, it was kind of a happy story, if you will, about something found on the walls of the subway station. And this then led me to one time I was describing these early results, and a reporter had said, well, Dr. Mason, can you tell me, is it okay to lick a subway pole? And I said, well, I gave a very long scientific answer. It depends on what's there, how long it's been there, the organisms, if someone came by that was sick. I said, but in general, it's mostly harmless, the things we found, and we can see there's even some good actors. And so, it's probably okay, probably fine, but, it really depends a lot on what's there. And then, of course, the only thing that got actually reported by the reporter the next morning was that licking subway poles is probably fine, says expert. Just to give you context of, if you ever talk to a reporter, just know that they can quote you down to 2 words and not care what you say the rest of the time around those 2 words. So just as a quick cautionary tale for the media engagement. But also, I put this up here because it's still true. It's 100% true that statistically, more often, most likely, you should be fine for what's there. And this is true just in general for what we found, but also we know this in general about how the body develops, that if you hyper sterilize your environment, you actually could increase your risk of allergies and other sort of complications with your immune system, because your body there's something called the hygiene hypothesis, which shows that your body, and especially your immune system, does need to develop with an exposure to the environment. So for example, if you think you're getting allergic to peanuts, you might think, well, the worst thing I should do is be in touch with peanuts because I'm allergic to them, but it's actually what you don't want to do. You need low level exposure is actually the best prophylaxis in order to avoid risk of allergies later in life. And this study has been validated now 3 different trials, and also is now part of an official therapy for peanut allergies, is you get little bits of the allergy to train your immune system. So it's just a different way of thinking about how your body reacts to sort of potential irritants and allergens. So that being said, this does not apply during pandemics. You generally still shouldn't necessarily, lick a pole. It's a mucous membrane is different from touching. Especially during a pandemic, you should not be licking poles. But you don't know what's there, so it's always good. But statistically, you probably should be okay. But still not always recommended advice. So that's a bit of the end of my social commentary and public service announcement. But okay, if you think fine. Thanks, Chris, for that weird non sequitur. But what else is there? What else did you find? So this led to a lot of excitement in the early days for the funding of the work, as well as partnership with the Bill and Melinda Gates Foundation when we first got it off the ground, and then also a global consortium that now works in concert to swab, annotate, and collect samples called MetaSub, the Metagenomics and Meta Design of Subways and Urban Biomes project, which is led out of Cornell and also with many collaborating sites, as I said, around the world. And our goals are to build out, these 3 things: these geospatial maps of the world around us, both metagenomic and even forensic maps, to trace antimicrobial resistance markers and look at both genetic and epigenetic differences, and also to look for new biosynthetic gene clusters, or really like new peptides, really literally new biology that's found on the surfaces of the subway poles. And so what we've been doing is actually since 2016, collecting thousands of samples every year. And you can see here, we've done, this year, Bob proud to include the springs and samples. I want to say hi. Just wait. Maddie says hi. This is my daughter. She's much older than she was in the pictures you can see here. I'm lecturing. Okay. Shut the door, please. And you can see here on Twitter also a lot of people using the hashtag to all work together and chat about what they're sampling. And what this has led to so far is some published papers and some in preprint, is literally a 3-fold increase in the size of the tree of life using these as well as some other data, and over 10,000 new viruses found on the subways around the world. And we've seen differences across the cities. So for example, how many cities have the most novel peptides? How many of them have completely unknown sort of organisms? And you can see here, Tokyo, for some reason, has the most novel peptides that we've seen from other cities for reasons that are not yet clear. But it's interesting, we see different cities around the world have different levels of novelty for what's in their biology. And we've also found, you can see here across these new viruses, what are called new CRISPR arrays. You've probably all heard of CRISPR and the ways you can use it for doing genome editing. So these are, new ways that actually the bacteria are fighting off the viruses. So these are the clustered, regular, interspersed, short palindromic repeats that give you the sort of a primordial bacterial immune system. And here, for type 1, 2, and 3, essentially CRISPR systems, we can see hundreds and even hundreds of thousands of different new CRISPR arrays, where this is evidence that these bacteria are actually fighting off viruses, just as we all are fighting off viruses every day in our body. Bacteria are doing the same thing, and you can see the evidence of that in the fragments of DNA that we pick up on the subway. And so this is another thing that's in the preprint. The other thing we've built is actually a map where you can type in any fragment of DNA. And if we know what species it looks like it belongs to, we can annotate that. But as I already told you, anywhere from a third to three quarters, and on average half of the DNA matches nothing known. And so if we don't know where something is or where it belongs or what species it's from, we can at least now map where it's gone. So we have this interface where you can type in any fragment of DNA and map the Kmer index, these sort of fragments of K length, so 13, usually 15, 17, or 19 bases at a time, and see where DNA is around the world and where it might be moving. And so, again, what's interesting is this seemed to be very abstract up until even January this year, but now the idea of having a global network that's tracing and mapping fragments of nucleic acids is exactly what's happening as people share sequence data about the coronaviruses that they're looking at. And so to that end, we actually, it's getting to the point where we're actually having planetary scale genomics, where we're, mapping and characterizing microbes, viruses, other organisms around the world. And that's the last thing I'll close with you on, is that what else do we need annotation for? What else is all the work that you're going to be doing helping with? It literally is helping with how we understand if there's microbes moving between planets. And so how will that be? Well, let me tell you. So we know that the goal is to get back to Mars, get to back to the moon, and then eventually go to Mars in the mid 2030s is NASA's explicit goal. We're not the only ones. There's now actually China and India are racing to get there. This is a picture from the Chiang 3, which landed on the far side of the moon. You can see here and set up some quick little hideaway. Some plants were growing last year on the moon, so it was a really exciting time in space biology to think about. You can get lunar plants. They died shortly after, so not yet, but at least they grew. There's even firms that have gotten funding from NASA to begin their moon-based projects. So literally, if you say, I want to grow up and be a lunar architect, that is a legit job in this day and age. And this is the kind of a rendering from one of the firms that won one of the awards of what you can look for. And so in that context, we're also taking swabs and sequencing things from the clean room. So actually from Jet Propulsion Laboratory with NASA, we have several projects with Katsuri Venkateswaran, who's a collaborator there, where we swab actually the rockets and also the probes and the rovers before they leave Earth and have their way to Mars. This is the case of the Mars 2020 rover, a component of it. And you can see here, there's Venkat. I put him up on the wall. But this room, it's supposed to be a clean room, but if you look around, it's clean, but not super clean, right? So if you look here, there's people standing around, and they, you've all been in the subway, so it's decreased transmission, but it's not like they're in BSL-4 suits. They're not hermetically sealed. So there is a little bit of engineer and scientist microbiome that's on the rover that's on its way to Mars in about 3 weeks. And so we've been, swabbing and sequencing the clean room and the rover to see what's there. And because people can't resist. Sometimes they'll take a selfie with the robot. I don't know if I could resist that either, quite frankly. But anyway, that happens. And so the question is, when we think about going into space and eventually sending probes, eventually this rover is going to make its way all the way over to Mars. There's actually a picture of one of the craters in Mars. And then in 2028, we're going to be getting a probe to pick up some of the samples, bring them back to Earth. One of the first things NASA is going to do is sequence the DNA and also characterize the samples in many ways. But they're going to see what's there. And one of the first questions will be, what are the microbes that look like they might be there, if there's anything there? Does it just look like microbes that came from the engineers or scientists working on the robot? Does it just look like normal Earth microbes? And in general, is there anything that could survive on Mars? And the best way to do that is to look at what's just surviving on Earth. Where are microbes found? What their sequence variation looks like? What sort of extreme habitats have they adapted to? And the more we understand the questions of the microbes that we found on Earth, the better we'll be able to answer questions about when and if things could survive on other planets. And there's even ongoing projects that actually Maria can tell you about, is to use some of this knowledge to start to engineer microbes that could literally on purpose survive on Mars and help with people that eventually will land there, hopefully in the mid 2030s. And so while it seems just doing annotation and looking at microbial facts seems very abstract, all that knowledge base gives essentially all the tools that we would want to be able to use for understanding biology, mapping where it's going, potential high risk, low risk areas, just fundamental understanding of biology, and even long-term plans to engineer biology so we could actually walk on craters like this on Mars. Okay, so with that as the intro, that's the background for the project, work that's happening in the lab. I want to thank funding from World Quant, the Gates Foundation, NASA, many NIH directors and divisions, and also thanks everyone in the lab who's made this all possible. And I want to hand it off to Maria and Krista to take it over. Oh, first I should thank the lab. This is a picture of the lab. We sometimes go out and go throw axes and drink beer, which sounds like a really awful combination, but you'd be surprised how well it works out. So anyway, I recommend this place in Brooklyn. It's a fun place to go visit as a lab. And that is it in a nutshell. And I'm happy to take a few questions before we turn back.
Again, my name is Maria. I am a team member of the Microbe directory. We've been working in this project for a little while. And basically, this project emerged in 2016 with Heba, who is also in the call, in 2016, and we collected about 7,500 unique microbial species. Some information that we collected of these microbe species were characteristics such as optimal pH, optimal temperature, pathogenicity, spore forming bacteria, biofilm forming, extremophile gram staining, and we compile all this data in a single website, which is called the Microbe directory, where basically you could type in any bacteria, any microbe that you are interested on, and then you will see different options. Basically, what you could see here is that you have 3 options. You will select the one that is the one that you're looking for. And then you will select Escherichia coli, which is the one that we're trying to find. And basically, you'll find this bunch of information in that might be relevant for a specific person looking for a specific microbe, or any large study looking to understand a microbiome. So this study was published in Gates Open Research in 2018, and it's called the Microbe Directory, an annotated searchable inventory of microbes and characteristics. And last year in December, we published a new preprint, which is the new version of the Microbe Directory, since we would like to expand it due to the acceptance of the public and the need for a reference database with microbial characteristics. So what is the relevance of this study? We know that it's a big effort to do manual annotation, but we wanted to highlight the key points and the relevance of this project. And first of all, this data is accessible and researchable through a single database. Sometimes when you are looking for a specific microbe, you'll see that you could find this information either complete or not complete, but in given databases, you can find sometimes all of the microbes you're looking for in a single database. This data is quality check, meaning that we've made sure that the data is correct and that it's reliable for you to use it in your study. Also, this, as I said before, this microbe directory is useful for individuals searching information about a specific species. So for example, let's say my mom, that she's not in the scientific field, but she's become very interested on the microbial world since all this pandemic. And let's say she wants to know about COVID, about SARS-CoV-2, she'll go to our database and she'll find that SARS-CoV-2 is found in certain hosts, and that if it has any other microbial feature, she will find it there in the microbe directory. But also, this microbe directory is useful for downstream large-scale metagenomic analysis, meaning that when the projects we have in the lab, we get basically a large list of microbes, but we would like to understand what those microbes are. Are they producing any compounds that might be relevant for the biotechnological field, or just a simple microbiological question. Are they gram-stain negative or gram-stain positive bacteria? All of this can be retrieved from the microbe directory. And finally, of course, we will be able to test hypotheses of microbial annotations based on these features. So for example, I have collected data of different environments. I'd like to see if there is higher abundance of certain microbes in my different biomes. You could test this hypothesis in the microdirectory. So we wanted to make the microdirectory as specific as possible for the different domains. And we have—
Hi, Maria. Can I just add in on that last part really quick?
Yeah, sure.
Hi, everyone. This is Krista. Just adding on to the purpose of the microbe directory, I think we've all been in a circumstance where we're looking for information online and perhaps that information isn't accessible. You need to subscribe, you need to buy it, and it's not free, or you're looking for information and you're not sure if it's accurate, you're not sure where this information came from. So a big part of this is allowing this information to be open source and open use for everyone, just purely for the purpose of education and research, and for it to be, as Maria said, quality as possible. Yeah.
Exactly. Thank you, Krista. And just to add there, sometimes we've seen volunteers really kind of worried and struggling finding available open source data, and sometimes they'll see that a specific database has the information about a given microbe, and then they'll tell us, oh, I know that that microbe is in that database, but I cannot see the data because it's asking me to subscribe and to pay subscription. And we want this data to be open and free for everyone in every country, everywhere. So this is a big effort, as I said before. But if you see the need, the necessity in every project needs to understand what the microbes are, and that's why this project is important, because every day we sequence more, we know more microbes, but we need to understand what is what are the characteristics
Yeah.
of these microbes.
Like what is out there and what is the baseline for the information on this specific microbe in general.
So we wanted to make the microbe directory as specific as possible for the different microbes. And we know that the microbial world is very diverse, and it was going to be really hard to annotate every single characteristic of every single species. So we went through the references, articles, and we tried to compile the most relevant characteristics for every domain, basically. So here we have bacteria, fungi, virus, and diatoms and algae. Now we are including diatoms and algae, which we've seen that there is a really a gap of information in these groups, and we wanted to include this in the microdirectory as soon as possible and try to give this data to all the scientific community, but also the public in general. So if we see more closely, we see that bacteria, for example, we are annotating certain characteristics such as gram stain, oxygen use, metabolism, and these characteristics, this bacteria also shares characteristics with fungi, such as spore forming. Is it a biofilm forming? What is the type of microbiome that you could find it on? Does it have any antimicrobial resistance? Is it a pathogen? Is it extremophile? If it's extremophile, what type of extremophile it is? And in the case of microbes found in the human body, which body side is it? most likely to be found. And in the virus, we have information about genetic material. Is it RNA, DNA? What type of a strand? Is it double or single? The capsid symmetry, the envelope, is it a pathogen or not? And what is the host where it's found? And for diatoms, we have an algae. Basically, diatoms are a big group of algae. So we have decided to split them and to characterize them more in detail. So for diatoms, we have this characteristics, but also some characteristics that are shared with the other groups of microalgae. So we have the lifestyle, if it's planktonic, benthic, what type of symmetry, secondary structures, antagonism, activity, is it motile or not? Does it make any symbiosis? Does it produce any toxins? What is the metabolism? And it shares some features with the algae, with other algae, such as the type of growth, the morphology, biofilm, spores, is it extremophile? Does it resist UV? Does it produce any pigments? What is the microbiome, the microbiome that it can be found? And for algae, we also have if it's a pathogen and what metabolites can it produce. So we've seen the pictures of the Lake Hellier in Australia, and it was actually an algae, and many people didn't know until we went there and we sequenced them, and we found out that it was an algae producing certain pigments that will make the lake look pink. So, Krista, would you like to add anything here? Anything I have missed?
So I would just add that right now, like this seems like a lot of information regarding your microbe, and that this may take a little bit of time to go into and find, or maybe you're just daunted because you're not familiar with the characteristics, the terminology. So, don't panic. We have gone extensively into defining all these characteristics for you so that you have a reliable source of information to, find what exactly is a biofilm. And we are providing you with sources so that you are not starting from scratch, just Google searching your microbe.
Yeah, we're gonna go through Coda, which has all the definitions for these characteristics, and also we have compiled for you a bunch of links and references, so you can go there and easily find the information. But it doesn't mean that you have to use only those. It means that it could be your baseline. So we are always expanding the micro directory, and our goal for 2020 is to have 80,000 species annotated, unique species annotated, with now more than 27 non-exclusive microbial characteristics. So it's a big goal, but we are pretty confident that working together we can achieve it. So this is, as I said, this is not easy. We are a big team. We have computer scientists in the lab who are the ones that help us analyzing and doing all the data management and curation. We also use some public databases to compile some data, such as NCBI, but we also need the manual annotations of most microbial features, and here is where your participation is key. It's very important, because without the manual annotations, we wouldn't be able to expand the micro directory. So we'd like to set a timeline in order for you to know how long you're going to be doing this. And we understand that some of you might be taking some summer classes, some of you have more time. So we want to do this for at least 12 weeks. It doesn't mean that after the 12 weeks you cannot continue. It means that this is the cycle that you will be participating on. If you want to continue, of course you can. If you want to participate in another project in the lab, you're more than welcome. But we want to acknowledge this your participation in the project, and we will give you acknowledgement on the publications, but also if your commitment during the weeks is you are always annotating, you are doing your best effort, but you are also doing a great, it's not only to annotate, it's not the numbers you annotate, it's the quality of data you annotate. And we see that you are really involved in the project. We'd like you to participate in the papers as a co-authorship, but you'll also be part of a consortium. And it means that whenever we publish a paper, your name will be listed in the consortium of the microbe directory there. So we know it's a big task, and we are really committed to help you to go through it, especially during the first weeks. It's going to be a learning process where you will start encountering some problems, like I cannot find this information, my species doesn't appear on the available databases or any article, and we want to help you. We will be there, but we also would like you to commit to 3 simple things, which is the constant weekly submission, which means that every Monday we'll send you the microbes, and then we'll require you to submit it before the next Monday. It's fine if you are, let's say that week you had some exams, that week you were busy with any family issue. It's okay, you can email us, and then we can make it work out. And the second thing is to make your best effort. As I said, it's not about the amount of microbes that you annotate, it's about the quality of data. You have to remember that this data is going to be used for other researchers. So we want you to take your time and to annotate genuine microbial data. And finally, to submit your citations, it's important to cite every website or article that you have used to get the information. We'll go through that. We'll go to how to submit your annotations, and your citations in a moment. We also have some channels for easy communication. We have Coda, which is the document where we have the microbe directory guidelines. We have the Kobo tutorial. We also have definitions for microbial features. And we have a Slack. I think most of you have signed up, and if you haven't, please do so. We share different useful links, different books, articles. Volunteers make quick questions or updates or any useful comments for other volunteers. Remember that sometimes you might have a question that other volunteer could have as well. So yeah, feel free to use this channel to share the microdirectory questions. And we also have the email, which will be used for personal emails, such as you cannot continue the project, or you cannot finish your weekly task on time due to any sort of inconvenience. You can email us there. We will try to answer as soon as possible. And I think that's it. We will go through KoboToolBox, which is the platform that we use to annotate data. And we'll also go through Coda Doc, which is the place where we have all the definitions for the microbial features we are annotating.
Maria, can I just add something?
Yeah, sure.
So hi guys, I didn't formally introduce myself. My name is Heva. I'm a researcher at the Mason Lab, and I'm a second year medical student at Weill Cornell Medicine. So we started the project in 2016, and it's grown and it's become like an amazing, amazing project led by Maria and Krista. But I think, so what I wanted to say is that it can feel very daunting as a task, and the first couple weeks it'll be kind of hard. There will be questions that people have where they're unsure of like what to put in the database, but we want to assure you guys that we're here for you to answer any questions that you might have, and it's okay to ask questions if you're unsure. In fact, we recommend it because odds are that someone else has the same question that you do. So for the first couple weeks, it could be hard, but we're just letting you know that we're here for you. And also, you'll find that as the weeks go by, you'll refine your own research skills. You'll have read a lot of papers, and some of our volunteers, like Hardik and Farida, if they're on call, can maybe speak to that. They've been amazing. They can maybe like speak about their experiences or what they found helpful or what they didn't find helpful. Another thing I want to add is that it is a work in progress. So we've been doing this for a long time, but there's always things that can be improved on our end and on yours. So communication is key. So do let us know if there's something. It can be just like a minor inconvenience for you, whether it's like data entry for something. But for us, but like if you tell us that this is a problem for you, we'll do our best to try and fix it. So again, please do communicate with us if you're running into any problems. It just go in with an open mind and be open to learning about the wide variety of microorganisms that we live with. So if Hardik, or I don't know if Farida is on the call, is she?
Rawa is also on the call.
Okay.
They could tell us a little bit more about their experience. They have been with us in the micro directory since February, January, I think. They've been doing an amazing job, and they all started as you are now. And some of you, as Heba said, might not have experience with this, and it's fine. We're here to learn together and to help each other. And maybe Hardik, would you like to?
Yeah, sure.
To give your personal.
Yeah, so it was actually a pretty cool experience. Like Emma said, that initially it was kind of like I was like, holy crap, this is like a lot of information. Like I don't know how I'm gonna do this and all that. But like as the days go by, you learn more, and it's crazy because over time I've actually seen my ability to analyze literature and just academic papers and just all that stuff, or even like just like databases and stuff, has really improved over time. So it actually gets easier over time, and you start putting data in faster and more efficiently, and also keeping up the quality, and sometimes even better quality. So it's super exciting also because it also has like opened up this like new door that I never thought about, like in genomics and even just microorganisms and stuff like that. And so like it's really cool. And like Dr. Mason was talking about it before, and just all these microbes and I guess like microbiomes that we have around us, and we really don't see them, but when you're actually inputting these datas and this data into Kobo, and you just realize that like, holy crap, there's more than like 2 million or even billions and billions of microorganisms around you, and you just learn more each day, and it's awesome.
Do you have a favorite microbe, Hardik?
Yeah, so I mean, with Maria and Krista and even Radwa, I've been working on like microalgae, and so I helped out a little bit with the Coda, and so it was just fun, like putting in just learning about microalgae. And it's amazing. They produce almost 50% of the oxygen we breathe, so it's important.
Thank you, Hardik.
Also microalgae, so
yeah, you have a background. Rawa, would you like to share your experience if you are out there?
So hi everyone, my name's Radwa. It was a really good experience. At first it was kind of hard because my first set of microbes were bacteria, and a lot of them had like very little research done on them. So it was hard for me to find information on them. And I was like, okay, great. I'm going to spend like 20 hours on my list every single week. This is absolutely fantastic. But eventually you adjust, you find sources, you learn like your own databases that maybe aren't on the useful links, which is something that I also wanted to stress. Like you mentioned that the page with the useful links is a good crutch, but I definitely wouldn't like depend on it, because in the process of doing your own research, you will find a lot of websites that you know you can trust and they have really good information. So definitely just, it's gonna sound weird to say, but definitely have fun with it. Like look for the different websites that you could definitely use. Setting up the extension with the microalgae and the diatoms is definitely a process, but it was also really cool to do just to see how useful they are in not just in what they do, but what they can eventually do. Like for diatoms, there's a lot of research into how they could help with like nanotechnology and biomedical use, and they're already used in like commercially or like industrially. It's honestly amazing to see. So yeah, you guys eventually will find your like your pace, your rhythm. It gets easier to annotate these microbes. It gets easier to find like reputable sources, and you're going to be a pro at it in no time.
I have to
Thank you,
ask
Radha.
Rawa if you have any favorite microbe also.
I mean, I'm partial to diatoms because those are the ones that I looked at, but I like looking at viruses.
Viruses are hard. Some of them are hard. Some of them you can barely find any information. Thank you, Radwa. I just wanted to also add that in our lab, so as some of you may know, I went to Hunter College, so CUNY, yay, if anyone's from CUNY. And I just want to emphasize, it can kind of feel like maybe we're all remote and there isn't a sense of community, but that wasn't the case prior to COVID. And also there is still a sense of community. So when I started this project, I was actually first recruited for the Metasub project just to do like a one-day swabbing kind of thing. Like I had no idea what this lab was about, and I was just going to swab some subways. Yes, Hunter College, yes. Camille, Dr. Chatterjee, been there. And then, so basically, I started out, just this project, and I was doing this swab, and I heard Chris talk, and I was like, this guy's amazing. Like, I really just want to be on board with whatever he's doing, right? So I ended up being a part of the Metasub projects, and I think the really, really great thing about the Mason Lab that is unlike any other lab really that I've seen is that even though like myself, I was young, I was just in undergrad, or I think what's different about the Mason Lab is that if you're really passionate about something, there's room for independence and growth where you can pursue projects if you want to do them, and if you're able to kind of grow in that sense, and you're given kind of the resources that you need and the support that you need from the lab to be able to pursue whatever you want. So there's definitely room for growth, whether after you finish this project there are other projects that you want to hop on board with, or other things. If you go like on the Metasub site or on the Mason Lab site and look at a bunch of different projects, and if any of them interest you, there's always room to do more. And we always do need people to do more, because sometimes I feel like we're stretched out way too thin. Like this guy's like in space and like in the Gowanus Canal, and he's like doing a bunch of different things. But he's an incredible scientist, an incredible human being, and endlessly curious. And I think that speaks volumes to the types of projects that we do in the lab, and your involvement and your commitment to it. So that being said, I'll hand it back over to Maria, but do let us know if you have any questions along the way.
I'm just gonna add something. I know we're getting sidetracked. Just to give some background, I think that a lot of people on the Microbe directory and even on the Microbe directory like team or leadership come from a wide variety of backgrounds. We're not purely microbiologists. Like I come from a background in computer science. Maria comes from more biology and then bioinformatics background. So, we are all, learning about this world of microbiology and also expanding our knowledge as we delve into this project. So don't worry if you are not a microbiologist. You know, we come from different backgrounds, and we are ready to learn more about microbes. And on jumping off of that, so if you are going through the data entry and you do notice that like an option doesn't exist where it should be and you are seeing inconsistencies, we definitely do take your feedback. We definitely are looking to expand the microbe directory more and more and be more all-encompassing to, you know, and making sure microbes are properly annotated. So, you know, definitely give us your feedback, and we will talk to you and respond and work with you to make this even better.
Thank you, Heba. Thank you, Krista. So we're going to go through the Coda doc that I think most of you have read already. We're going to discuss some important things on the Coda doc. Can everyone see my screen? The Coda doc is basically a document where you can find different tabs, and each tab will give you certain information. So for example, in this one, which is the guidelines, you'll find all the kind of the frequently asked questions of the microbe directory. What is the microbe directory? What will be my task as volunteer? Where is the KoboToolBox? Some important notes when searching. So I'd like to emphasize here, sometimes, as we have said, you will not find information about your microbe, and it's important to know when to use NA or when to use unknown. So if you, for example, find no information about anything, you cannot even find an article or a database with your microbe, you could even send us a question on this Slack and tell us like you haven't found any information about a specific microbe. We'll go through all the resources and try to find information. But if after a while you haven't found any, you could select NA or unknown. So some examples of when to use it is, for example, let's say you have a microbe and you found that that microbe does not produce biofilm based on the reference. So the reference says this doesn't produce biofilm. You will select no as the answer of biofilm forming. So then if, for example, the same question, you are looking at the biofilm forming characteristic and you cannot find whether the microbe produces the biofilm or not, you will select unknown, because it could happen that it hasn't been studied by researchers. So it's fine to select unknown when you don't find any information. And if you find that certain characteristic does not apply to your microbe, so for example, such as biofilm in virus, they cannot produce biofilm by itself. So you will select NA, since this characteristic, this microbial feature does not apply. But there is another case when you can select NA. So for example, your microbe doesn't seem to have a species name. It says unclassified, or it says, for example, unidentified protobacterium or unidentified marine eubacterium. Sometimes this could happen, and this usually happens when the studies cannot classify to species level due to some to sequencing issues, or there is not enough information about the genome or the sequence of the microbes. So these microbes are not annotated to species level. So here, for example, if you're annotating biofilm formation, you cannot know if the microbe of unidentified protobacterium produces biofilm. You cannot find it. You cannot know because you don't know what is actually the species. You will select here NA. Is that clear, or is it too confusing? in this.
Wait, I have a question, Maria.
Yep.
So, like many times it comes up like, oh, it's like unclassified, and then it gives like like how it has a taxonomy, it'll give the one level up, and many times you can find a lot more information on that, right?
Yeah.
So would you recommend that to the volunteers or no?
So it could happen that your microbe is classified to genus level. So genus level is a higher level from a species level. So the genus level could give you information, and we have a section here that you can annotate, annotating at genus level. So if you cannot find your, sorry, if you got a microbe with only the genus name, you will annotate characteristics that apply to all the members of the genus. So for example, if you found that Streptomyces produces spores, all the Streptomyces produce spores, so you will select, okay, spore forming, yes. But if you find that certain characteristic can change within the genus, can change between species. So for example, the Gram staining. So you find that Streptomyces could be Gram stain. Some species can be Gram stain negative, and some species can be Gram stain positive within the same genus. You cannot annotate here based on the genus level. You will have to annotate NA in this case. Is that clear now? I put some also some other examples here on how to annotate to genus level. You can read it more carefully after, and if you have any further questions, feel free to use the Slack channel, and we'll address your question as soon as possible.
Maria, if I may add one thing, sorry. So I just want to emphasize how important it is to read the Coda document, like really, really, really thoroughly, because so as a research team, we're all collectively putting out information out there, right, for the scientific community to use, for the public to use. And to this day, this project's been out for 4 years, but to this day, we still get emails. What did your research team define as a biofilm, right? What did your research team define as this or that? So it's really, really important that we're all on the same page. We all have the same understanding of what we're annotating and what the characteristics are. And there's going to be discrepancy in terms of, what other scientists classify as something, or what other research communities classify something. So it's really important that you go by the definition that we set for the entire project as guidelines. And it's really, really important that you take the time. Like if there's one thing that you're going to do, you're just going to read this document, make sure that you understand everything, and that it hopefully gives you a clearer idea of what is expected. And if you do have any questions, again, feel free to let us know.
Thank you, Heba. Something important you mentioned is that, we still have getting emails about some other researchers. How do you define here biofilm forming? So again, it's important for you to read this document, so you know what we mean with biofilm. You know what we mean with pathogen. I'll go through the pathogen option very soon, because here sometimes there is a confusion about what is a pathogen and what is not. So I'll continue quick here. Where to look. As we said, we have useful links, which is right here. You could find it also here. And basically, this is a table where you find a bunch of databases and some websites that we have carefully selected for, and we know that you are more likely to find some information about the microbes. But again, if you want to use Google Scholar and find information in research articles, we'll encourage you to do so. Sometimes you will find in this table that, for example, for bacteria in this database, you can find Gram staining. It doesn't mean that 100% you will find that. It means that it could be there because they annotated, but it could be that they didn't have that information for that particular species that you are searching. Moving on, we also have some cases where you will find in the names of the species. For example, you are looking at Streptomyces, let's say Streptococcus mutans, let's say. So Streptococcus mutans is the species of bacteria that usually lives in the airways. So Streptococcus mutans, you go through the databases and you'll find that you find your species, but with Streptococcus mutans 1, 2, 4, and you'll see like what is that? What does it mean? It means that the species that you found is at a strain level. So for example, here I have a table, an example of this bacteria that we are annotating. So you'll find sometimes in your database, in their databases, your species with certain characters at the end. This means that all of these are a strain level. And in the microbe directory, we're not annotating for now a strain level microbes. We are only annotating to a species level. So what are you going to do if you find this information? So you're looking at Abiotrophia defectiva. So you could use the information in any of the strains as long as it's consistent within the other strains. So for example, you click here, this one, the first one, and you see that this bacteria is gram-stain negative. And you click here, oh, again, gram-stain negative. And you see here, again, gram-stain negative. Okay, this is consistent, it's gram-stain negative. But if you found here, for example, oh, this one has antimicrobial resistance, that you go to the other ones, all of them, you don't have to go to all of them, but you click multiple, and you found that this has no antimicrobial resistance. And then you go to the articles and you cannot find any antimicrobial resistance in Abiotrophia defectiva. So then you're gonna be like, okay, what's going on? Because they found one strain that says that it has antimicrobial resistance. This could happen when a particular scientist is interested in Abiotrophia defectiva when it is expressing antimicrobial resistant metabolites. So they will modify the microbe in order to make it resistant. So it means that this particular strain was used for a specific study to make to be antimicrobial resistant. But the species itself, it's not antimicrobial resistant. You know what I mean? So sometimes some strains might have some information contradicting to other strains. So just make sure that the information, whenever you are getting information from a strain level, is consistent among all the strains of the same species. Is that clear so far?
Yep. Yep.
Yes.
Okay. So for example, again, as I said, sometimes your microbes might come as unidentified or unclassified, and that's fine. You will basically, if you find, for example, this, you find this is not a species name. This is a microbe that was found in another animal or another plant or another host, and but you don't have a specific species name. You will annotate again NA because it cannot be annotated based on without the species name. We had some questions during the first cycle about synonym species, and sometimes, for example, when researchers go through one species and they sequence again and they do a phylogenetic tree, they found that they might be clustering different, and just because before we didn't have all the techniques and the molecular techniques to correctly build the phylogenetic or the tree of that species. So it could change the name due to reclassification. It doesn't mean it's a different species. It means that it was reclassified. It was moved from this part of the tree to this part, but it's the same species. So if you find a specific synonym, a species with a synonym, you can use the information of the synonym as long as it's consistent. You find, for example, that this species was first described and then it was changed to this species later that year, and then like 10 years or more, yeah, even more than 10 years, was reclassified and was named now like this. So it's the same species again, but it was reclassified due to new technologies. You can use the information in any of the synonyms as long as it's consistent. So again, you can go through this in more detail, and if you have questions, feel free to reach out to us. Again, I already explained the annotating at genus level. You can annotate characteristics that apply to all the members of the genus. And if you're unsure about the microbe that you have the species name, but you don't know whether it's protobacteria or if it's an algae or diatom, you're not sure, feel free to go to NCBI taxonomy, where we get all the list of species. Here, I'll click here, you will find here basically all the species that have been submitted in every study around the world. So basically you will type on your microbe and the taxonomy, let's say, for example, this one. All the taxonomy of your species will come up. So for example, this is the genus Arabidopsis, which is a plant. And you will find all the, like the domain, the phylum, kingdom, and so on. Just so you have clear what is the taxonomy of your species. Another question that we get very often is, what is the difference between microalgae, macroalgae, microfungi, and microfungi? How can we differentiate? So when we got the list of NCBI, we get me the list of all the fungi. You cannot retrieve a list of microfungi and macrofungi. What do we mean with micro and macro? Basically the size. So macrofungi will be like the mushrooms you may have in your fridge, and the basically the mushrooms that you will see in the forest or anywhere. And microfungi means the fungi that you need a microscope to see. You cannot see it with your eyes. Basically, you need a microscope. So same for algae. So sometimes in your weekly task, you will get some macro fungi and macroalgae. We are not interested on that as for now. We are only interested in the ones that are microscopic, because microbe directory, right? So just make sure that your algae or fungi is, what is it, if it's a micro or a macro. We have a question in Kobo where you have to select if it's a micro or macro, and we'll go through that in just a minute. So finally, okay, how to cite information. We recommend this link, which is so thorough. Basically what you do is you paste the link of your website or your article, and it will give you a reference like this. And basically what you'll do is copy and paste in the empty box you'll find in Kobo. So you will copy this. So for example, let's the microbe directory. And you'll find here we have the Heba Shaban article, and you will copy it. You copy this and you paste it. That's it. It's pretty easy. Here is an example on how your references should look like. if you find information, multiple answers to multiple questions of the same species in just one reference, you can annotate, you can citate it only once. You don't need to citate it like, okay, I use 5 answers 5 times the same. No, you only need to do it once, okay? And basically that's it. These
Maria,
are,
sorry, can I just add something?
yep.
Sorry, just regarding that point. That being said, a lot of volunteers, including ourselves, when we did this, we found it a lot easier to not close your tabs, because sometimes you might find like one piece of information and input it in the directory, in the database, and then close the tab, and then you're like, oh, what was it again? What was the paper that I was looking for? So make sure that like all your tabs are open until you cite everything, and then once you submit for that one particular species, you can feel free to close them. That's one thing. The second thing is, as Krista said, we want this to be a resource that people can have access to. So that being said, there shouldn't be an article or paper that could have answers that you shouldn't have access to. So I know that through a lot of colleges, like through their libraries, like through Hunter or Brooklyn or the CUNYs, you can access a lot of these articles through the college's library. But if that isn't the case and you're still not able to access it, you can send us the link and we'll try to find it, download it, and send it to you. So we want the database to be a centralized, curated database. So for example, when Dr. Mason and I yesterday were swabbing and people came up to us and said, what are you doing, right? Like, we just look so weird with like our little swabs, and we're sampling and our little gloves on and everything. And we had to explain to people, you know, we're collecting samples and we're trying to figure out what is it exactly that lives on the subway, right? So when we do that, take it back to the lab and sequence it, and we get a list of these species, whether they're viruses or bacteria or anything that live on the subway. We want our database, the microbe database, to be the database that researchers go back to and say, okay, I have this entire list. I just want to know, to get like a profile about what is in the, what did I find in my sample, and for it to be centralized and to have as least discrepancies as possible. So that being said, if Maria, as Maria mentioned, if there is a discrepancy between 2 sources that you find, maybe consult a third source or consult us or use your judgment. And you may be a little hesitant to do that in the beginning. And that's normal, right? But you'll find that over the weeks, you'll use your judgment a lot better. So I'll just give like a quick example. It's better to be, I guess, more general than more specific and inaccurate. So if there are 2 sources, one of them says that something is an obligate aerobe, the other says it's just an aerobe, and you're not sure, and you check a third source, but you can't put your life on it and say it's an obligate aerobe, then it's always better to put that it's just an aerobe. Does that make sense? So use your judgment in terms of like what could be more accurate, because remember, we're putting this information out there for people to use, and we want it to be as accurate as possible. So yeah.
Okay. Thank you, Heather. And we'll continue very quick through the Kobo definitions. You'll find here the definitions that for the questions that we try to annotate for every microbe. And if you click, for example, virus, you'll find what is a virus, the graphical representation of a virus, what is genetic material, and the type of strand. What do we mean with capsid or naked, and the capsid symmetry. What is a pathogen? So, and also some useful links here if you want to go through more specific research articles, and the type of hosts, and some questions will be similar in the different groups. So for example, pathogen, it's also present in bacteria, fungi, microalgae, and diatoms. And I want to mention here in the pathogen, I'll go through this first. So for example, sometimes you will find that your species, your bacterial species, was isolated from a patient that was immunosuppressed, for example. So usually we're not going to annotate bacteria in specific cases where the patient is immunosuppressed or the patient was sick, and they found the particular species that was present there. We're going to annotate pathogens as if only the species that whenever we're in contact with, the host becomes sick. So for example, let's see here, Staphylococcus. So as I said, Staphylococcus is always present in the skin microbiome, the airways, or airways in the human body. And sometimes they have found that Staphylococcus, which is the normal microbiota, can become pathogenic when the host, when the person becomes very sick or immunosuppressed. Here, Staphylococcus is not considered pathogenic because it's usually found in the normal microbiota. But, for example, Mycobacterium leprae. So this bacteria is a slow-growing bacteria, and whenever the human is in contact with this bacteria, they cause leprosy or the Hansen disease. So whenever in contact with this Mycobacterium, the person gets the disease. So this is considered a pathogen. Here, we're only considering pathogens that are not within the normal microbiota. And this applies to humans, plants, any host, animals, and so on. Is that clear for now? Okay, so here definitions: what are bacteria, what are archaea, the Gram staining, and what is a biofilm, an example of a biofilm, the oxygen use, again, what Heba was mentioning, when is anaerobe, when is, it could be an obligate anaerobe. Sometimes in the articles, it is not going to explicitly say the answer, but you can infer it through the article and through the results of the article. The metabolism, sometimes this might be a tricky feature, because in order for the researchers to annotate the metabolism of a given microbe, they have to isolate it. And as you may know, not all of microbes can be isolated or are easily isolated in the lab. So for you to, for a researchers to understand what is the metabolism, either photoheterotroph or phototroph, chemotroph, anything, they have to isolate it. And this information is going to be available mostly for microbes that have been isolated. So don't worry if you don't find the specific metabolism. For example, you could find that it's a phototroph, but you don't know what is the electron source or the carbon source. So you will select phototroph. Let's say you found the energy source and electron source, but not the carbon source. So you will select photoorganotroph. Is that clear? If you cannot find anything at all, just select unknown for that given microbe. We selected some examples. Okay, and spore-forming pathogen, I went through it, and the antimicrobial resistance. Something it might be important here is that the resistance, the ability to resist any antibiotic or any antiviral, antifungi, it's totally normal in nature. It occurs every single day since day one of life, basically. But the process of like the humans taking antibiotics and using all the time sanitizers and cleaning all these microbes could speed up the process of developing resistance. So here we are annotating those microbes that didn't have antimicrobial resistance before, and now due to our activities, they do. So here, this is what we mean with antimicrobial resistance. And extremophile. So extremophiles are actually, I think, my favorite group, but they are sometimes difficult to annotate because they are not totally well studied. Here, extremophile is basically a microbe that can thrive and they're not normal conditions. We say normal to what's normal for us, for human beings. But for them, the extremophiles, let's say a bacteria that lives in really, really, really acidic environments. So for that bacteria, that environment is normal. Of course, it's not. We are the extremophiles for them. So in that case, the extremophiles the microbes that live under really not normal conditions, such as acidic environments, really hot environments, really cold environments, they are called extremophiles, and they can be different types. And here is the description of the different types, and they could be sometimes multiple types. So for example, if you look at the Yellowstone Park, you will find hot springs that sometimes are very alkaline or acidic, but they are also very hot. So there you will find microbes that resist high pH or really low pH, but also thermal conditions. So that would be either acidophile or alkalophile and thermophile. And finally, the microbiomes, different microbiomes. If you find any microbiome that doesn't fit within these answers, these options, feel free to contact us and tell us that your microbe lives in another totally different microbiome that we haven't included here. All right, and microfungi, again, what is the difference between macro and microfungi? And an example of a spore-forming microbes, fungi. Again, the pathogen, antimicrobial resistance, and the microbiomes. And okay, and microalgae, I'll head it up to Hardik. Are you around there?
Yeah, I'm right here. Right here.
Okay.
All right, cool. So let me also open up on my side. Okay. So microalgae, yeah, it's a unicellular photosynthetic organism. It can be facilitated heterotroph, but we have a type of growth, so we can either have solitary or colonial. So if you can scroll down, morphology can be important. We have different shapes and sizes. You will also find some that might not pertain or might not come under this chart. So we'll definitely have options for that. And like I was talking about metabolism before, they're mostly autotrophs, but they can also be facilitative heterotrophs. And there's also something really cool about microalgae. There's something called mixotrophs. So they literally use photosynthetic components and heterotrophic components to improve their outcome, because microalgae are used to harvest. They created a bunch of byproducts that are really important. And also, if you scroll down, we have pigments. And yeah, so there's a bunch of pigments, chlorophyll, and then you got beta carotene, and you'll probably find much more. We also have mobility, so like non-motile and motile, and you'll probably have to annotate what type of mechanism they use for mobility. And then we also have biofilm. Biofilm, it's just like bacteria. They're used to provide an environment for microalgae to thrive. And reproduction is through spores, and it can be either asexual or sexual spores. So yeah, and it's very important is also symbiosis. Like microalgae, there's a known symbiosis between coral and microalgae, where coral provides an important space for, or like a safe space for, microalgae to thrive. Pathogenicity is also present in microbiology. You'll find some, it's few. You might find some invasive microalgae. Really just depends. It's a little bit rare, I would say. But what's really important, I would say, is biotechnological applications. So I think the biggest thing microalgae has going for it, I mean, besides providing almost half of our air, is their byproducts. So it literally produces lipids, proteins, carbohydrates, isoprenoids and terpenoids. That's like an organic chem molecule that if you take an organic chem, you'll know about. They're actually precursors to like big synthetic molecules. You kind of like put them together and they do like a reaction, they form a ring, and you could create a bunch of rings with them. So, and they produce toxins. Toxins can be important because they can be actually antimicrobial. So they can actually be used to kill other microbes, so which is also important. UV resistance, some microalgae have the ability to grow in very harsh, I guess like sunlight, like if there's a lot of sun, and they have the ability to resist the UV. And extremophiles, Maria already talked about. But yeah, that's pretty much it. Yeah.
And as we said, diatoms are a big group of microalgae. Rawa, would you like to jump up here and explain very briefly what the diatoms are?
Yeah, sure. So diatoms are kind of like a subset of microalgae, so there's going to be a lot of overlap between the things that we're going to need to look for microalgae as well as diatoms. So a lot of it is similar. So as you can see, there's same question of type of growth, of whether it's solitary or colonial. A lot of diatoms are in aquatic habitats, so there's also a question of lifestyle, of if they're free floating or if they have some sort of substratum they're attached to. And depending on the substratum, they're annotated differently. There's different symmetry of diatoms, which is just either pennant or centric. Same for microalgae. There's the question of morphology, which is super important because it helps with their classification. There's definitely feel free if you feel like you found something that isn't listed. That happened a couple times in our cycle. I know it sounds like when people say, yeah, you can definitely ask questions, you're still scared to ask questions. But as someone who just went through this as like a new volunteer, they're very nice with questions. Like they'll answer all of your questions, and you won't feel like you're a bother. So definitely ask them. If you find something that's not listed in the Kobo questionnaire, then you can literally say, hey guys, I found this and it's not one of the options, and they alter the questionnaire. They're super nice. So for diatoms, their cell wall, the frustral, that's a common characteristic, but some of them have secondary structures like spines or bristles. Again, if you find something else and you think it's not on the document, just let us know. There's also biofilm formation. Same with microalgae, there's UV resistance, antagonism activity, locomotion. Same pigments that are being formed, they have different functions, whether it's photoprotective function or just aiding in photosynthesis. Same with symbiosis. Diatoms are usually, if they do have a symbiotic relationship, it's usually with cyanobacteria, but occasionally there's also symbiotic relationships with other microalgae and maybe coral. Again, whatever, if you ever find anything that's not there, just say so. Some diatoms have toxin production, and another common question among all of the microbes is just extreme affinity and metabolism and microbiome.
All right. Thank you, Rawa. And I want to mention here the useful links. Feel free to go through all of this and check all the available references. If you find any good reference that we haven't listed here, please let us know so we can include it. And we're gonna go through KoboToolBox really, really quick. So we have a tutorial here in the KoboToolBox in case you want to go through it in more detail. Krista, would you like to jump in?
Yes, of course. So KoboToolBox is an online data collection tool. You can use the Coda here to learn how to log in to your account. Account will be provided to each of you as well as a password. So don't worry about registering. But when you do log in, a good thing to note is that there are 2 logins for the site. There are the humanitarian organizations login and the researchers and everyone else login. And when you do log into your account, make sure you click researchers, aid workers, and everyone else, or else your login and password will not work. They are 2 separate sites, so just be careful. And as I said just earlier, you don't need to create an account. We will provide you with a username and password. This is just so that we can keep count of your entries and make sure that everything is secure from the back end. If you do feel like creating an account for your own use, I do recommend the site. It's very useful for data collection. And as you log in with your provided username and password, you will see a deployed form like this. Yours will say the Microbe directory data entry form, volunteers summer through fall. And you will be able to click that and you will see a little tab on the bottom that says collect data, and then next to it on the bottom right, it will say copy or open. And you'll just simply open that form and it will go into your entry for your single microbe. As Maria stated earlier, you'll be assigned 10 microbes per week. So you'll simply need to copy 1 microbe from your list and paste it into the scientific names box. I'm actually going to share my screen really quickly and just show you what the form looks like live. Let's see.
It is important that you copy and paste the name of your species because sometimes you might miss any character and it will not match within with our records. So
Right.
we will see that, oh, this person didn't submit the microbe, but in fact they did submit the microbe. It's just that it was something small character difference.
Yes. So thank you, Maria. As she just said, just copy and paste it. The microbes you are sent may have asterisks, they might have brackets or parentheses. Just copy and paste it as such. If you do notice that your microbe has multiple different types of names, some have a purely scientific name, and then others will also have a, I wouldn't say modern name, but alternate name. I'm trying to think of an example. Like, I believe, like the chickenpox virus.
Are you sharing your screen? I can't see.
Oh, you can't see it?
Yeah, I can't.
Just as example, the chickenpox virus is also called varicella. So if you do see there's an alternate name, you can always share that information with us. So I'm going to stop share and share again. So in this tab, you'll see what your actual form will look like. There will be a few tabs. You don't need to worry about that. You'll just need to press open and you'll open to a form like this. You will actually be able to see all information that you have submitted. So after you've been submitting microbes for a while, a data tab will appear and you'll be able to see all the information pop up. So you can definitely verify that your information has been submitted and verify all of your records, just in case you are concerned that we are not receiving your information. So you'll see a form like this, and again, I said you just need to copy and paste it, and then you'll just need to specify your domain, whether that is archaea, eukarya, or a virus. We do ask that you answer these questions in sequential order. And that's just because we are the entry form is dealing with the branching of logic. So if you press virus, you'll get a different set of questions. If you are pressing eukarya, if you're pressing bacteria. And you'll see all the questions that Maria went over with the Coda. It's not loading right now, but it should be fine. Viruses, different set of questions, and eukarya. So as Maria mentioned, we're dealing with different types of eukarya, whether they're fungi or algae. If you are assigned a fungi, you have to first specify whether you're dealing with a microfungi or a macrofungi. Because we are at the microbe directory, we are looking solely at microfungi. So if you are assigned a macrofungi, you can simply fill in the information as such and submit your form. Don't worry that it's short. You'll just have one short entry for the week. And then the same for algae. You need to specify whether it is a micro or macro algae. And macroalgae would be like seaweed or something that is more visible. And if you are assigned a microalgae, you should specify whether it is a diatom or not. And you'll get a different set of questions. So after you have entered all your information, make sure you go through each and every question. As you can see, there's a little asterisk next to some questions. That means that you have to answer the question before you are able to submit the form. So just make sure you're very thorough. This question, I'm just going to go over specifically because it's a little confusing. You need to specify whether the bacteria is is photo, chemo, organolitho, heterotroph, or autotroph. So as Maria showed in the Coda, you're going to click, if you can, 3 of these options. And if you find that something is a photoautotroph, but you cannot figure out whether it is organo or litho, just enter as much information as you can, or it's a chemo autotroph. So I don't know if Maria, you want to speak more to this or?
Yeah, I think you can annotate the information that you find. It's fine if you select unknown, because again, the metabolism of every microbe is not well known. So some of them are going to be there, but some of them not. So yeah, annotate as much information as you want, and always remember to use the citation empty box, which is right there.
Yeah, so we do have the link to ZoterraBib here, and we have the text box here to enter all your citations. You just need to copy and paste them into the text box, and you don't need to copy and paste the same citation over if you're using the same reference for multiple questions. And then just please enter your full name. We do keep track of your entries using your username, but sometimes there are some errors in capturing your username. So just enter your full name just so we have multiple accounts of your entry and that you do get credit for this information. The form does work offline, so if you do find that your internet connection is unstable, all information will be saved onto the cache and your actual computer browser, so don't worry about that. If it goes through, you should be able to see an entry that says successful submission. If you don't see that, just please reach out to me. The Kobo interface actually just had an update about 2 weeks ago, and there's been some glitches, but it's mostly been quite reliable. I do recommend that you use either Safari or Google Chrome for submitting KoboToolBox entries. Other browsers are fine if you have one that you want to test out, but those seem to be the most reliable. Are there any questions? Yeah, and we'll get your credentials to you before that so that you can log in and you can go through the form and become more familiar with it.
You're not just limited to these characteristics that you're annotating, right? If there's something that you find interesting, go ahead, take some time to read about it. So like medicine and microbiology go hand in hand, immunology. So like right now in medical school, we're learning about like all these different ways that these pathogens and microbes can sort of evade our immune system by acting dormant within our cells, right? Or for example, if you know that the bacteria that causes tuberculosis that was once like a deadly disease, if you know that it's like an obligate aerobe, which it is, it's not hard understanding why it predominantly attacks the lungs, right? Where there's always oxygen. So it always goes hand in hand. Environmental science, there are pathogens that are really, really terrible for the environment, but there are also microbes that can help us degrade plastic or degrade hydrocarbons or help us with pollution. So it goes both ways, right? So just I guess try to lay the groundwork this week, because your future self will thank you when you're doing the annotations. Take the time this week to sort of go through the characteristics and learn more about them, and feel free to, you know, consult like other sources, watch YouTube videos if there's something that you're still having trouble understanding the concept of what it is we're trying to get at. Do feel free to contact us, but make sure you do that this week. Just take the time to go through each one and understand basically what is expected. But also we understand on your part that people, you know, not everyone has the same experiences. Some people have more experience in microbiology than others, and we'll try to do everything that we can to make sure that we provide resources to help everyone, and, you know, to help get everyone on the same page.
Thank you, everyone. Please make sure you log into Slack so you we can keep in contact. And again, we'll send you the credentials by the end of the week. And please make sure you read the Coda doc just to make sure all the definitions are clear by Monday. If you still have questions, please reach out to us. Thank you very much for your interest, for being here these 2 hours. And we'll see you during the week and during the following weeks. Thank you, everyone.